What we're going to do now is talk about the digestive lab, which means we have to introduce some processes of the digestive system. Obviously, we're going to really introduce it formally next week. So we're just going to talk about the stuff that we need for right now. And, it's not going to be a surprise to you all that the digestive system is about breaking down your food and absorbing the nutrients, right? Digestive system. And food is usually just the body of some organism, right? It's some plant or animal that had its atoms and molecules assembled in whatever it needed to be assembled to make a cow or a apple or whatever. And then you take it in your body and you break it down, like basically into building block molecules. Right? Those amino acids that used to be part of the cow or used to be part of the carrot are now disassembled into individual amino acids that you can then assemble into whatever you want to make your body, or maybe break them down for energy. All of those carbohydrates that were stored away by that potato for a rainy day, you've disassembled into sugars for your own energy. All of the fatty acids that were creating the fats that, helped insulate that pig for, when it's out in the pasture or whatever, you've disassembled all those into individual fatty acids that you're going to use for your own body and your own cell membranes or whatever. So that's a big part of the digestive system, breaking down your food into these building block molecules, and then, the absorption. Absorb, and we're going to look at this in more detail in class, but for this lab, it's looking at the breakdown. It's looking at the breakdown, and there are a variety when we talk about like food nutrients, and we talk about fats, proteins, carbohydrates. These are the main nutrients that we can actually break down for energy. Other things that we'd call nutrients would be like minerals, which are just elements like calcium or phosphorus or sulfur or whatever, or vitamins, which are essential, like kind of organic molecules that you have to get from your diet. Sometimes people even say water. But we're going to focus in on these. So specifically, where this lab looks at specifically breakdown of fats, proteins, and carbohydrates. And we're going to look at it in vivo, not in vitro. What does it mean to look at something in vitro as opposed to, versus in vivo? Does anyone, what is vitreous? What does that word mean even?
Glass.
It means glass. So this is like, in a test tube. We're basically going to be, ultimately your fats, proteins, and carbohydrates are breaking down in your body, in your mouth, or in your stomach, or in your intestine. But it's a lot harder to manipulate the conditions and see like exactly how does pH affect things or this or that if it's all happening inside your body. So instead of looking in the body, we can create these artificial conditions in a test tube, in a glass, and it just means in glass outside of the body. And the nice thing is you can really control lots of things, concentrations of different things, whether or not, what is the pH? Is there going to be bile salts in there or not? Blah, blah, blah. So, I mean, the bad part of in vitro is it isn't real. It's not going to, there could be complexities going on in the real body that you're missing if you're doing this kind of artificially controlled reaction in a test tube, but it can give you a lot of good information. And that's what we're going to be doing in this lab. We're going to be looking at the protein, breakdown of fat, protein, and carbohydrate, in test tubes, and where we can change the conditions, see how pH affects things, things like that. So that's kind of the big picture. What I'm going to do is go down. Actually, I'll start with carbohydrates, then go to protein, then go to fats, and talk about the specifics. What is our example for that? For carbohydrate, we're just going to use starch. For the protein, we're going to use egg white. For fat, we're going to use cream, milk cream. And we're going to talk about each of these, talk about how they're broken down in the body, and then what we're actually going to manipulate for our actual experiment. So let's start with carbohydrates. Let me double check time here.
If you woke up feeling tired today, keep watching the video.
What the heck? Hold on. Hold on one moment. There it goes. Okay. And what the plan is going to be is I am going to kind of lay this out for you. Then we're going to go into breakout rooms and just like we did for the kidney lab. You can work together and make sure that you're all coming to a consensus on your hypotheses. And there's going to be another thing where there's going to be lots of mini parts to the hypothesis. There's going to be like 15 different test tubes, and each test tube needs its own hypothesis. So I want you to kind of work on that all together in the breakout rooms. The thing that's nice, this is the last lab. There is no lab after the digestion lab. So you are going to come up with your hypotheses together. Next week, we'll actually look at the data and see what we found, and interpret that in the context of our hypotheses. But we have made it to the end of labs. So let's do carbohydrates.
Are you gonna want us to turn it in after class?
Yeah, you should. Yes, there's a link for just for the hypotheses, though, not for the whole lab. So for carbohydrates, we're going to use starch. Starch is just lots of glucoses connected together in big long chains. Right? We talked about starch in the first week of class. It's like the main storage carbohydrate for most plants. And we're going to use a starch solution. We're going to use starch, dissolved in water. It makes a colloid, which we saw in the first lab. So we have starch. We're going to need a way to assess starch breakdown. So in this case, we're going to have two different tests. Lugol's iodine tests for starch. Basically, the iodine is kind of a brownish amber color. This basically stays kind of amber if there's no starch. Oops. So if you have starch and you put a drop of Lugol's iodine in it, you'll know the starch is in there because it's going to turn kind of purple black. If you put a drop of Lugol's iodine and it just stays, it's normally kind of this ambery color, and it stays kind of ambery, then you know there's no starch. So that's going to be one way to assess. If we put starch in the tube and we add iodine and it turns purple black, we know there's still starch in the tube. The starch did not break up and go away. If it is amber, though, then we know that there's no more starch left, so it must have broken down. We also, in this particular, for the carbohydrate assay, we have an assay for the breakdown product as well. What do you get if you break starch apart?
Glucose?
Yeah, you get glucoses. If you take it all the way down, you get glucose. The enzyme that we're going to use from your saliva is called amylase, or salivary amylase. It doesn't break it down to individual glucoses. It more breaks it down into shorter chains. So for instance, this would be a maltose. Maltose is like 2. So when we do the more formal discussion of the digestive system, we'll talk about the enzymes in the brush border that do the fine cleaving of disaccharides and the monosaccharides. But we're going to be looking at breakdown in the mouth and with salivary amylase, and we're going to end up with maltose. And there is a stuff called Benedict's solution. Which is for maltose, or it's just reducing sugars. Stays blue if none. Turns different color. If there's in the presence of maltose, it can go from green to orange to red, depending on how much. So this is kind of nice. We have starch. If I have starch and it hasn't broken down at all, then glucose, there shouldn't be any of the simpler sugars in there. So that means Benedict's would be just stay blue if there's been no breakdown, and the Lugol's should be purple black because there's still a lot of starch left over, right? If the starch has broken down, then Lugol's is gonna not find any starch, so it's gonna stay amber, but my Benedict's is gonna start turning green or orange because there's lots of maltose showing up. So we have 2 different things to test for the breakdown. Like in your hypotheses, you need to give me a result for both Lugol's and for Benedict's, what is happening. In terms of the actual lab itself, let me get my. So an amylase is just an enzyme that breaks down starch. There's lots more starch breakdown that happens in the small intestine. We're not worrying about that. For our lab right now, we're going to focus on the very first steps of starch breakdown, which happen in the mouth. Again, the actual enzymatic breakdown is going to happen from this amylase, which we're going to get just by spitting in a test tube. Literally, you spit, it's in your spit. And what are the conditions in your mouth? If we think about what's the temperature in your mouth?
Close to body temp.
It's going to be like body temperature, right? So when we actually incubate, we're doing things in vitro, but we're going to make sure we put our test tubes in this water bath that keep them near body temperature. So we're mimicking the conditions in the body. What is the pH in your mouth? Because remember, enzymes are very sensitive to pH, or else they're going to denature, right? What would be what's the typical pH in your mouth? Or most of your body, unless you're looking in weird places.
Seven.
Yeah, around 7, around neutral. Remember we said like, your blood is around 7.4 or so, but in general, things are pretty neutral. So that's going to be important. We're going to be playing around with pH, and that's going to be one of the things that you're going to have to think about when you're coming up with your hypotheses. So we have these test tubes. In the first test tube, we're just going to have starch and water. And we're going to let this sit, at body temperature for like a half an hour and see what happens. Another one is going to have starch and saliva. We're going to let that sit for a half an hour at body temperature. The last one, we're going to have concentrated hydrochloric acid and starch and saliva. And let that sit for a little bit of time, half an hour. Does anybody have any guesses? What would we expect if you just left starch sitting with water for a while? Would you expect to see much breakdown?
No.
No, there's no reason. Starch is pretty darn stable. If you have some spaghetti on the shelf and you come back a week later, it's still just sitting there. It hasn't changed much. What about starch and saliva?
It should break down because of the amylase.
Exactly. The salivary amylase we just talked about, its job. And as long as the conditions are correct, its job is to break down starch into simpler sugars. What about this one, test tube 3, that has the concentrated hydrochloric acid?
Won't that denature the saliva so it won't be effective?
Yeah, right. Because if we all of a sudden have a really acidic environment, what's going to happen to our poor enzyme that's been optimized to work basically at neutral pH? That's not, yeah. So when you're coming up with your hypotheses, think about that. Think about this idea that you have this really acidic environment and what happens to an enzyme if you take it outside of its pH comfort zone. And this is going to be a little different. When we look at protein digestion in the stomach, there you're going to have enzymes that like to work in really acidic environments. So they would want it to be more acid, but salivary amylase is optimized to be in the neutral environment in your mouth. So this is the basic experiment. Like in your hypotheses, you just need to say for test tube 1, test tube 2 and 3, what is the Lugol's and the Benedict's results going to be for each of these, and why? Is it going to be negative, meaning I didn't find either starch or sugar, or positive? So the other thing we do in this lab, we do have a test tube 4 and 5 as a control. Let me just talk about that super briefly. This is just going to be starch and water. This is going to be maltose and water. And the reason we do this is going to be basically to validate our assay. Right, we're trusting. I've told you, like, oh, the Lugol's does this, or the Benedict's does this, but you want to make sure they're working properly, making sure your tests are working properly, that your reagents aren't contaminated in some way you're not expecting. So when you do an experiment like this, it's always a good idea to use something where you know the answer, and then double check that you're getting the answer that you should be getting. So if I took test tube 4, in fact, for both of these, let me just So what should Lugol's tell me if I am in test tube 4 with just pure starch and water?
That it'll be purple or black.
Exactly, purple or black. I should just get positive starch. Benedict's, what should I get for Benedict's?
It should be red.
Red would mean that there's maltose around.
Blue.
It's going to be blue, meaning negative. There's no maltose. For test tube 5, where I have just pure maltose and water, that's where I'm going to have a positive for Benedict's. But Lugol's, what am I going to find for Lugol's?
Amber.
Amber, a negative test. So this is this kind of validating. If I do this and I don't get these results, I can't trust any of the other ones I'm not sure what's going on. So you do this to make sure that you trust your test. I think
Sorry, go ahead.
No.
Oh, I just wanted, should we be predicting what color the Benedict's would change to as well, or just that it will change?
You can just predict, because you can't really know. Yeah, you can just say like positive or negative.
Okay. And then do we need to do hypotheses for 4 and 5 because they're the controls?
Yeah, because
Okay.
right, because if your predictions aren't correct, that means that you have to go back and figure out what went wrong with your whole experiment.
Okay.
You should definitely have a prediction.
Okay.
But those predictions are like, they better be right. The other ones might or might not be right. But I might have mentioned, like, back in, I think it was the 70s, when people were really like, people were publishing things saying LSD breaks people's chromosomes, all these scare things. And, when people actually went back and looked at those studies, they found that what they lacked was this kind of validating of the assay. Like in the test that they did, when they added the LSD solution to the assay, it came out positive for breaking chromosomes. But when they added, milk it also tested positive for breaking chromosomes. When they added tap water, it also came out positive for breaking chromosomes. So, so obviously, whatever this test was telling you was not that interesting at all. So, you always want to put something in the test where you know what the answer should be. If you put tap water in the test, it should not be breaking chromosomes, So that's where this validation is important. Because otherwise, when you're trying to get the answer to something you don't know whether you actually trust your assay. If you have yesterday, it was actually it was an article, it was 2 days ago in the New York Times, all about the latest phase 3 clinical trials with MDMA. That's all been more and more positive stuff. There's a big picture of my friend's wife in there, which is kind of fun. All right. So any questions about carbohydrates? All right, let's continue. Proteins. Our protein in this lab is going to be egg white. Egg white is just albumin. So cooked egg white, that white rubbery stuff, is just a bunch of tangled up protein. Protein is initially broken down in the stomach. By an enzyme called pepsin. Pepsin basically breaks apart the peptide bonds. And we haven't really talked about this much. We have talked. What's the pH in the stomach like?
Really low, acidic.
It's really low. pH 2 around. So this enzyme that we're talking about then is going to have a pretty different pH that it is optimized to work in, right? Pepsin likes it to be really acidic. That's where it shines. pH 7 would be like denaturing for something like pepsin, because it would be really alkaline compared to what it likes, where it has the perfect shape. So that's important to think about when we're coming up with hypotheses here. Think about where pepsin is optimized to work and what kind of pH it is actually looking for. The assay for the protein breakdown is going to be a lot more qualitative. It's basically going to be take your egg white and cut it into little squares. Each square is going to be 7 millimeters on a side. So they're all about the same size as much as you can. And then you're going to put it in the test tube with the different stuff. And after an hour, you're going to look. And if the protein has been digesting, meaning the peptide bonds have been breaking and the amino acids just dissolving into solution, if I look at a square that's been in there and it's like that big, it's like, oh, I guess as we've been digesting, because all the rest of that protein has now gone into solution. So our assay for this one is just going to be look at the size. Compare where we know they all started exactly the same size, so then we can tell if it's breaking down because it'll be smaller than that. And on this one, We have 5 test tubes. Yeah, they all have egg white. So they all have a little square of egg white in them to start. You know, test tube 5 is just going to have H2O. Now this one's basically going to be our control. If you have egg white just sitting in water, you're not going to expect anything. So we're going to be able to compare the other ones to this piece after it's been sitting in the incubator for a while. Test tube 1 is going to have pepsin in water. Test tube 2 is going to have HCl in water, hydrochloric acid. Test tube 3 is going to be pepsin and HCl, hydrochloric acid. And test tube 4 is going to be pepsin and sodium hydroxide, which is a strong base. Make it very alkaline. So again, just from our discussions about optimal pH and all that, which one of these tubes would you expect to have the best protein breakdown?
3.
Right, 3. This is what's mimicking the best what's going on in your stomach. Acidic environment along with the enzyme to break down protein. You know, that's what is actually going on in your stomach. What about what's going to happen in test tube 4?
The least breakdown,
Right, and why?
because the strong base will negate the role of the pepsin.
It's going to like denature it, right?
Yeah.
Yeah, and similar here, pepsin and water. Here, this is kind of neutral. Maybe it's going to work, but definitely it's not going to be the ideal pH that pepsin likes to work in. You know, so when you're coming up with your hypotheses for these, you know, I would say put it through the lens of pH and what pH pepsin normally works in and what is the pH of the other tubes. And then HCl, hydrochloric acid, and this is going to be a weak acid, or it's a weak concentration of a strong acid. But acid is going to kind of denature things. It's not necessarily going to break things down, right? It's going to kind of tweak the bonds and stuff and unwind things, but isn't necessarily, in terms of actually coming in and chopping apart the peptide bonds, that's something more that you need some enzyme to do. The enzyme can actually get in and break the peptide bonds between the amino acids. So that makes sense? Yes, no?
Yes.
Yes. Okay. The last thing we're going to talk about is the lipids or the fats. You know, this one we're just going to use kind of cream as our example. You know, and cream is already not just a pure fat. If we were going to use a pure fat, we'd be using butter, but then it's really tricky to kind of mix it with things. So cream is already got the little fat kind of distributed through something more watery. So what we need to talk about is the idea of emulsification. So let's talk about emulsification. So let us say you just took a swig of olive oil. All right, so we have olive oil. and it's inside. You've just swallowed it down into your stomach where it's all kind of watery. What do we know about the glob of olive oil sitting in a bunch of water?
Nothing happens.
I mean, well, nothing, something happens. You know, it stays in a big glob, right? It's hydrophobic, so it does not go into solution. The big glob stays a big glob, separates, right? You've all seen this in, if you've ever made just a salad dressing with vinegar and oil, and you shake it up, and then the vinegar and the oil separate again, right? You know, so the problem with this, you might have your enzyme. In fact, we do have an enzyme. So this is going to be, is the the breakdown is, this is going to be in the small intestine is where we're going to watch the breakdown for this. There's an enzyme called pancreatin. That is a lipase, something that breaks apart lipids. The problem with this lipase is even if it's floating in the water, I'll actually make it red so you can see it better. I'm making it like a little axe trying to break apart the fat molecules. It can't get at most of the fat molecules because they're all in the middle of this big fat glob, right? And if I shake things up, I could shake it up into lots of little droplets, but before you know it, they're all gonna glute back and become a big drop again, right? You've seen that. You shake up oil and water and it separates again. So what we want to do is some way to shake this up into little droplets, but not have the drops all come back together. And that's going to be emulsification. And it's going to be done with an emulsifier, in this case, bile salts. So basically, you know, one side polar, other non-polar. Right, amphipathic. You've seen this, like with the phospholipids that make up a cell membrane. So what happens is you basically shake this up into little droplets. And now the bile salts can get in. The non-polar side goes into the fat droplet, the polar side goes to the water. So now the droplets don't have this drive to re-glom together into just one big drop, because each one can float around as an itsy bitsy droplet. So that's what emulsification is. It's, you're not really dissolving the fat in the water, but through the help of these bile salts, you are able to have the little droplets stay distributed through the water and not re-glom together. And what that also means now is my enzyme has way more surface area to work to break down things. Right here, I could only get at whatever fat molecules were right on the outside, but now I can get at each of these little droplets. I got so much more access to the little fat molecules to break them down. Right, so pancreatin, a lipase, it can break down lipids on its own, but if you use bile salts along, you are going to have so much more surface area to access. It's going to be so much better, right? Does that make sense? So just as we should say cream is already slightly emulsified. If you didn't have that, if you didn't have some emulsification going on there, it would just be like butter. So obviously the fact that it's like a half and half that you can pour means that there is already some emulsification happening in just the cream itself. But if we add bile salts, we can even make it more emulsified. So the only other thing I need to add in here is I didn't talk about how can we test for the breakdown of breakdown of the fats. Do people know what is litmus? Is litmus paper, litmus powder? The litmus test. What is litmus testing?
pH.
pH, right? Litmus is blue and alkaline or neutral. And it turns kind of pink or red in acid. So when we first add litmus powder to cream, it's just a neutral pH. It's just going to look blue. It's going to look like blue cream. What happens when a fat, remember what is a triglyceride actually made out of? What does a fat actually look like?
The glycerol and 3 fatty acids.
Exactly. So if you start breaking this puppy apart, you're freeing up the fatty acids, which means things are going to get more acidic, which means it's going to turn pink. The more breakdown you got, the pinker it gets. In fact, in this assay, you can go from blue to like lilac to purple to, to red. So basically, the more fat breakdown you get, the more acidic things get because you're freeing up more fatty acids, and the pinker or redder it gets. So that's going to be your test when you're trying to figure out if fat breakdowns happened. If it just stays blue, the fat didn't break down because there's no fatty acids that are free. The more breakdown you got, the redder, because fatty acids broken away makes it acidic, makes it pink. And then if we look at our test tubes for this one, it's going to just be 4 test tubes. Everyone is going to have the cream, the litmus cream. This one will have cream and pancreatin, which is our lipase. This one's going to have cream and bile salts. This one's going to be cream and pancreatin and bile salts. And this is just going to be cream and water. This does not say cream. So again, what would we expect here? Which one do we think is probably going to have the best breakdown?
3.
3. And why?
Because you have the bile salts that will make the oil drop smaller into smaller droplets, and the pancreatin will be able to break them down more efficiently.
Exactly. Right, this like if you end up getting like your gallbladder removed, you're going to have to be really careful about your fat intake because you're going to be so much worse at breaking down fats because that bile the bile salts in the bile from your liver, from your gallbladder, is what really makes your fat breakdown efficient. What about test tube 1?
Some breakdown, but not a lot.
Yeah, so we've got the lipase, so we're probably going to have some breakdown, particularly because I said cream is actually emulsified to some degree. But, definitely it's not going to be as efficient as if you're increasing the emulsification by adding bile salts there. What about this one? Cream and just bile salts. Are we going to expect any breakdown? A chemical breakdown of the fats?
No.
No, because remember, bile salts don't actually break apart the molecules. All they do is allow the little droplets to stay little without having to regroup. But you're not actually going to be breaking anything down. You're not going to break a triglyceride into fatty acids with bile salts. They don't do that. They don't get into disrupt the actual bonds in the molecule. They just go and disrupt the bridge the polar nonpolar divide between the water molecules and the fat molecules. And then cream and water is just, again, it's going to be more like a control, right? You're not going to expect the cream to do much. Water in general is pretty passive here. So maybe let me go share. Hold on. Modules. Let's share. So here we are, Digestion Lab. This is the very last thing. And if you look at the for salivary digestion of carbohydrates, there are basically 5 tubes in all. For each tube, you need to have a prediction for your Lugol's iodine assay for starch, as well as your Benedict's solution assay for maltose. For gastric digestion of protein, we have 5 tubes, all with egg with varying pHs and absence or presence of pepsin. For each of these, you should have, again, this is more qualitative, like no breakdown, or maybe a little breakdown, lots of breakdown. So you want to just have a prediction and a Y. Remember, obviously, each of these needs to have a Y as well. And then digestion of fat with pancreatic lipase and bile salts. Again, 4 different ones, like we just talked about, some with cream and bile, some with just cream, bile, and pancreatin, some with just pancreatin, some with just bile, and one with neither even. Right, so for each of these, in your lab notebook, again, it can just be a short thing of what you expect in terms of the breakdown and why. So are there questions about that?
So do you want 3 objectives and 3 sources of variability as well?
Sources of variability, I'd say you don't need to redo for each one. That's going to be, the sources of variability are kind of similar for the whole lab. So I would say I'd say one sources of variability for everything. And what are major sources of variability in a lab like this?
Contamination.
That's a huge one. In fact, that's one that some kind bites us in the butt, where if somebody's like dipping the wrong pipette in the wrong solution, and we do the I said validate the assays, and it's like, holy crap, there's there's maltose in this pure starch. So we know, like, dag nabbit, somebody put the maltose pipette in the stock starch solution. So that means everybody's starch actually had maltose in it to start with. So yeah, contamination's a huge one. Yeah, so things like that. So, all right, so it's 12:14. You don't have to finish, but I would like you to get started together because I do think this kind of thing is easier, if everybody's kind of talking to each other. And some people kind of get get it quicker than other people. That's just the way it works. And then it's really nice to have people kind of explain it to each other. And again, the more you explain it to other people, I think the better you get at it. And then having the person who gets things explained to them, they don't stay confused and they have someone to help guide them along. So I'm going to shut down the breakout rooms at 12:30. But try to get as far as you can, I would say. But again, an individual hypothesis for each tube, kind of like we did for that RBC lab. Each tube should have a prediction, like what you expect and why. And again, it might just be a sentence. It doesn't have to be that elaborate. Most of these are not going to be that elaborate, but it should be clear.
The hypotheses, should they relate to each other like they did in the osmolarity lab, the kidney one? Like for each section? Like this one's going to be the most reactive, least reactive, that kind of thing?
Or some of them might be like nothing. Yes.
Okay.
Yeah, I'd say it's not necessarily. Yeah, it's definitely doing something relative might, particularly like for the protein one, would make sense. For some of the other ones, that might not make as much sense.
Okay. And also objective for this lab, we're just doing one objective or objective for each section?
I'd say no, because each one, like if we look at salivary digestion of carbohydrates, what are the independent variables? What are the things we're varying between the different tubes?
Saliva and concentration,
Well, what do you mean by concentration?
the HCl.
Right. What is HCl? HCl is concentration's not so clear. What is HCl altering?
pH.
Yeah, pH, right? So HCl, so we're altering the presence or absence, and saliva in more, what's exactly is saliva giving us that we really care about?
Amylase.
Amylase. So we're going to have the presence or absence of the enzyme amylase, and then vary the pH between kind of normal and really acidic. Those are, and our dependent variables are going to be the presence or absence of starch and the presence or absence of maltose. So that the objective for that would just be like, how does, amylase, and pH affect, the breakdown of starch and appearance of maltose. For the protein, instead of, here actually it is the enzyme as well and pH that we're altering, but instead the dependent variable is going to be the breakdown of the protein. For the fat, our dependent variable is the breakdown of the fat, but the independent is going to be enzyme or bile salts, right? So your objective should just, in a nutshell, make sure it encapsulates what exactly we are manipulating and what we're looking at. Again, it shouldn't be more than a sentence. And next week, we'll look deeper at this. But for this first part, I just want you to kind of get your, kind of think about what we're doing and what you expect to find. And we'll look deeper at this on Tuesday after our quiz. All right, so it's now it's 12:19. I'm going to go let's see. Here, we'll just do 3 so there's bigger rooms. And we're going to create these right now. And again, talk amongst yourselves. Make sure you reach a consensus. I think it's always an interesting process to, like, if somebody's kind of lost or something, or isn't, I'm not sure I get that. Try to make sure everybody don't. One of the problems that can happen is when you get like somebody who really understands it well in one of these breakout rooms or in a lab group, and they just kind of keep plowing along and leave everyone on the dust. So make sure that everybody kind of understands before you kind of move on to the next part. And I'm going to create now, and I'll see you actually in like 10 minutes, and we'll just kind of, you're not going to finish in 10 minutes, but you can get started. And then we'll kind of just say goodbye. All right, so it's another 10 seconds and everyone's gonna get kicked back in here. Here they come. All righty, so any questions about any of these little hypotheses? And I'm happy. What I can do is, if you want to just finish this up while you're here, depending on what you've got next, I'll make some of these breakout rooms again that you can just join in. You can tell each other or chat with each other which room you want to go into. That way you can actually just finish this up if you'd like. Minimally, you should be turning these in, before the lab. So like on Monday is when they'll be due officially, since you need to have these before we do the lab on Tuesday. But otherwise, we're pretty much done. So what I'm going to do is I'm going to make breakout rooms for people, and then I'll also be in office hours and discuss things with people for office hours. And I hope you all have a good weekend. And yeah, what else? It's Willie Mae's 90th birthday, which is kind of cool. Yeah, when I was a kid, I used to go see him at Candlestick all the time. It was really fun.